And as the other, at least some of the other speakers did so, I would also like to congratulate the whole consortium to reaching this state. And I would also like to thank, in particular, Yolanda Sands for being part of this consortium, both on behalf of myself and behalf of the colleagues that are still back in Copenhagen. So my talk is going to be on one of the specific work packages in the MyNewGut project, specifically looking into the effects of fiber supplementation in the treatment of obesity management. And I will also present some data on a meta-analysis that some of my undergraduates were doing as part of the project. So I'll present two lines of evidence. So one is the MyNewGut Work Package 8 randomized controlled trial. And another is this systematic review and meta-analysis looking at the effects of dietary supplementation with fiber. Our aim was to look at the combination of inulin and resistant maltodextrin as a supplementation of 20 grams of fiber per day. And we decided to use maltodextrin as the placebo treatment. It was a 12-week intervention with a hypercaloric dietary regimen, so asking the volunteers participating in the project to be actually undernourished in terms of calories during these 12 weeks. We did the intervention in overweight and obese people, and we were looking at a number of different outcomes, obviously changes in body weight, but also body fat percentage, cardiovascular risk factors, glucose homeostasis, and so on. And then at the same time, we had some students to look into the literature to look, okay, so maybe there is some findings to be done in our own project, but what if we compare to the current literature as we have been looking at? They kind of undertook the same assessments in terms of what kind of studies would they like to include, and I'm not going to go into detail with this, but obviously it had to be randomized controlled trials. It has to be fiber with at least some similarity in terms of composition and dosing, and study duration should also be reasonably comparable, and it should also be done in overweight and obese populations. The data that I'm presenting today is unpublished, and they might be subject to change. I don't think it's going to be major changes, so if you have some good comments today, I'm open for suggestions. So more into detail with this project, it was a randomized controlled trial, and the population was obese, as said before, BMI 28 to 45, adults, non-smokers, and then we had this energy restriction as part of the intervention. We expect, although we don't have the data available today, that the habitual underlying fiber intake in those individuals would in average be around 24 grams, maybe a little less, could be a little bit more. And then the fiber supplementation was this mix of 10 grams of inulin and 10 grams of resistant maltodextrin that Natalie just showed was some of the fibers that are indeed fermentable also in humans. The placebo group was 20 grams of maltodextrin, and the study design is shown here. So once you know that the participants are eligible, they were enrolled for 12 weeks of randomized treatment, not knowing if they had the active fiber or the placebo fiber. And so the most critical part is here that we do clinical examinations at the study entry and then at the end of the 12-week of treatment period. During the intervention, they get regular dietetic counseling to enforce the caloric restriction. We had about 116 that were screened and just eligible for randomization. During the intervention, we had some dropout, ending out in a total of 86 study participants to complete the final 12-week clinical assessments. There were no differential dropout between the study groups, so no adverse effects leading to differential dropout. And these are the results for the baseline clinical assessments. Using a randomized design, you would hope and you would also expect that you have reasonable comparable groups. This seems to be at least somewhat the case. So this is the fiber group, this is the placebo group. So this is the baseline characteristics of those that actually completed the whole 12-week intervention. They were fairly well matched in terms of age, also in terms of most of the other outcomes, but actually they did differ somewhat in terms of body weight. And that's probably, or that is at least partly based on the fact that you cannot 100% sure be able to randomize into two identical groups, even though we did randomize for gender as well as body mass index. But there seems to be a little bit of slight differential dropout in the two groups. So we end up with a slightly heavier group in those that received the placebo treatment. In terms of the results, which is obviously the most interesting part here, I guess, we found that, as expected, that we would induce a weight loss. So if you compare baseline to 12 weeks, both for fiber as well as for placebo, you would see a 5 to 6 kilo of weight loss in average in the two groups. But in this case, we don't see any between group differences. So even though they actually all get a significant weight reduction, lowering waist circumference, lower fat mass, lower body fat percentages, and so on, the changes are comparable between the two treatment groups. In terms of some of the other clinical outcomes that we looked at, such as systolic and diastolic blood pressure, lipid levels in terms of changes in HDL, LDL cholesterol, and so on, as well as glucose homeostasis, we don't see any major differences between the groups. They all have the beneficial effects from the weight loss that you would expect looking at diabetes and cardiovascular risk factors, but we don't see any between group differences. There are some changes that might survive a P threshold of less than 0.05, such as changes in systolic and diastolic blood pressure. On the other hand, when we look at the fasting insulin levels, it actually seems to go in favor of the placebo group. So we have to be a little bit cautious of interpreting the between group differences. We also looked at some of the liver or inflammatory parameters, such as liver enzymes and hsCRP. No difference between the groups. We have some potential bias in the sense that when we try to record physical activity during the intervention using 7-day accelerometers, we see that the placebo group was slightly more doing slightly more physical activity during the intervention, and we will obviously try to adjust our results accordingly. But it's not going to make the huge difference to the results that are already presented here. But that's shown in the latter part here. So PA vector is actually a measure of physical activity using accelerometers. And it was slightly more active in the placebo group. So a cautious interpretation of the dietary supplementation study for work package 8 is that we do not see any between group differences in terms of the anthropometrical parameters and glucose homeostasis. We might see some small effects in terms of changes in blood pressure, but we'll have to dig into more statistical analysis in order to say that more confidently. We also did some search into the literature using what we believe is kind of standard practice for doing meta-analysis, and see. So these are some of the tools that you would apply. I'm not going to go into detail with this, but basically we search for literature investigating the effects of dietary supplements with fiber. Not necessarily looking for changes in body weight, but at least also recording changes in body weight and other metabolic outcomes of interest. And we looked at different outcomes. This is just showing the plot for the studies that investigated changes in body weight. And so we were able to find 16 studies with a study design, fiber supplementation that was within the criteria that we had defined. You would see that most of the studies are finding an effect in favor of fiber. So if the dots are on the left part of this line here, then the effect is in favor of the fiber supplementation. If you look for the absolute changes or the absolute differences between the groups, you would have to look into this column here. So the studies find anything between basically a small gain for having dietary fiber into something that leads to minus 16 kilos of body weight loss in the favor of dietary fiber. And just to make some comparison, being an obesity researcher and trying to follow the drug development. So the most promising drugs today that are approved includes at least some of the GLP-1 analogs. Some of those products usually give an additional weight loss of about 4-5 kilos in a year. Orlistat, which is another weight loss product, is giving an additional weight loss of about 2 to 3 kilos. So even finding a, say, natural or at least a harmful dietary supplement that could give an effect of, say, 2 kilos would really be quite impressive, at least compared to pharmaceutical drugs today. So if you add the studies together, the average is 1.8 kilos. If you look at some of the other study outcomes, they seem to be somewhat aligned with the changes in body weight. So we see improvement in some of the anthropometrical parameters, so body weight and body fat percentage, also glucose homeostasis. If you then decide to specifically remove one of the studies, in this case a study by Gente et al. In 2009, because it's kind of an outlier, then the estimates are still in favor of the fiber dietary supplementation. Again, the effect sizes are not so large, but they are still, I would say, at least statistically and possibly also clinically relevant. They also try to dig into whether the type of fiber would have an influence on the overall effects. In this case, it tried specifically to look at the interventions that either looked at viscous fiber or non-viscous fiber, so no combination of fibers. And in this case, as you can see here, it seems that the viscous fibers may be more effective in terms of introducing weight loss. But of course, the sample sizes are relatively small. It's 4 studies and 4 studies respectively. They also try to investigate whether the study design would have an influence. In the MyNewGut work-paid project, we decided to have a hypocaloric regimen. You wouldn't necessarily need that in order to test whether dietary fiber works. And in the literature, you would find that if you specifically look only at the studies without caloric restriction, then you would also find a significant effect from fiber supplementation. I think that we should still be a little bit cautious in interpreting the data here. First of all, it's very important to stress that the studies out there are very heterogeneous, both in terms of the type and the amount of fiber, the study duration, in terms of the clinical outcomes that they are using. I I guess we still need more and better studies, and obviously I cannot rule out that publication bias is also at play here. We don't know that. So sum up, just some discussions or some issues for the future. Obviously, looking at the literature as such, we would need to consider if we can truly compare or merge studies with very different study designs and very different types of fiber that's been applied for tests. And also for the MyNewGut Work Package 8 study, we are curious to see whether we are able to distinguish characteristics of responders versus non-responders. Could be that some had some phenotypic features that would make them more inclined or likely to actually have a positive response to dietary fiber supplementation, but we'll have to look into more detail with that. So with these words, I will say thank you for your attention, and also thank you very much for the whole consortium for being here today and to present the final results. It's really been a pleasure to be here. Thank you.
So thank you, Thomas. One or two burning questions. Natalie, yeah.
Yeah, so maybe, yeah, thank you. So thank you very much for your these. So as a medical doctor, so there are a lot of medical treatments also in those patients and so on. Do you think that rather than thinking about having a supplementation in patients with no treatment, it would be maybe better to see if these type of intervention could be helpful in the improvement of the drug effect and so on, not only on obesity but also on other parameters?
I'm not fully sure I understand the
We can think about having this type of approach in patients to try to make them lose weight and so on without taking into account their drug treatment. Do you think it would be more relevant in the future to think about, I would say, a helpful effect of this intervention together with the drug treatment in the patients?
Obviously, I guess there are some combinations of treatment that wouldn't go together. So surgery and probably some other measures at the same time wouldn't go. But in this case, doing combinations of drugs, I guess there are already ideas out there with the poly pill and so on. So combining drugs as well as dietary supplements, I would not rule out as a possibility, but of course it has to be tested whether there are really additive effects from doing that.
Thank you so much for the very nice presentation and work. I was curious what type of immunogen you had used, like the chain length, and could you comment also a little bit more on the type of resistant starch, whether it was 2 or 3 or... Thank you very much.
That's a very good question that I unfortunately is not able to answer here. That I will have to look into the details of the study. Yes, thank you.
Then we have another question on the right-hand side. Yeah.
Thank you, Ina Schulten-Weissman from the Translational Microbiome Center at the Karolinska Institute. What was your scientific rationale for choosing those fibers, and how do you think about setting a dose? Because I think that is extremely difficult.
In this case, it was mainly a decision based on the fermentability. So we were quite reassured that these fibers would certainly be something that we would be able to ferment in human bodies. Obviously, when we don't have the data for the microbial composition that we will have eventually within the next couple of months or so, then we'll be able to see whether that criteria is actually really fulfilled. But of course, fibers are very different, and as I indicated, the effects maybe even on colon bacterial composition, but even also in, I don't know, in appetite regulation, in postprandial glucose regulation and so on, maybe different fibers have completely different effects. So in some of the outcomes that I've looked at here, maybe it would be more efficacious to look at other types of fibers.
Then I have one last question. So Do you know what was the final fiber intake in the test group, given that your background diet was already so abundant in fibers, which I think is quite typical for a study population in Denmark?
Yeah, I would expect, and we had some data from the other project that was done in Denmark, that the average fiber intake is probably around 24-25 grams, but it could differ in those particular participants, and there might also be some kind of range. So some might have, I don't know, 15 grams, and others will have 50 grams, I don't know. So in average, I would say 25 grams. We still have to analyze the food diaries that were collected during the study, also to see if there were major differences during the study intervention.
Okay, thank you very much.